My name is Clifford Brangwynne. I'm a faculty member at Princeton University, and I'm the co-director of the physiology course. I've been here many summers for many years, including as a student in the course, a teaching assistant, rotation faculty, now co-director of the course. I've also been a Whitman scientist and generally have a deep love and affection for the MBL. One of the memories that is most exciting in my experience in the MBL was in the summer of 2008 when I returned as a teaching assistant at that time, I was a postdoctoral fellow in Dresden in the lab of Tony Hyman, who had been a faculty in the physiology course for a number of years. And so we came back in the summer of 2008 and brought C. elegans worms, and I had just started studying this problem of P granule organization. The basic idea is that you have these C. elegans embryos, and P granules are RNA and protein-rich assemblies that are initially scattered throughout the C. elegans embryo, and then over the course of about 10 to 15 minutes, as the embryo polarizes and sets up for cell division, the P granules all collect in one side of the embryo. And in the course of imaging this early developmental process in the C. elegans embryo, we were looking with a couple of the students, David Corson and others in the course, and we noticed that some of the P granules, when they were collected on one side, they were very round, these kind of round structures that we hadn't really thought about that so much before. And if they bumped into each other, sometimes they would fuse and form a single larger round structure. So we were making movies actually right here on the third floor in the Lerb lab at one of the microscopes. And we started scratching our heads and saying, gee, it's kind of interesting. They almost look like droplets. That conversation just kind of spiraled. And then a lot of things suddenly snapped together. It's like a puzzle, so you get one piece and then a whole bunch of the pieces snap together. Because the segregation process that we had been studying suddenly made a lot of sense. If they're droplets, well, then you could regulate their assembly and disassembly very easily by condensing the droplets over here and then dissolving them over here. And then we started wondering what the properties of these droplets would be. And from the fusion measurements, you can estimate properties like viscosity and surface tension, or at least their ratio, and you get a sense for that. So that's really one of the most magical things for sure that's ever happened in my career, and it happened right here in the physiology course, which has always been for me it's just so foundational to my entire career, and now we and others are involved in efforts to find drugs that target droplets in disease contexts, and there's going to be drugs that going into clinical trials sometime this year and next that ultimately trace back to these very basic observations in a curiosity-driven and based course here at the MBL, which is, I think, something really exciting and almost gives me chills. I think the physiology course is immeasurably impactful, and the reason I say that is because there are so many students who come through the course who, they reflect back on their experience in the course, say that this has been the most transformative educational experience of their lives, of their careers. Many of these students go off in the world and they bring the spirit and culture of the MBL, of the physiology course, out into the world. And that spirit of fearless interdisciplinary approach to fundamental scientific problems, I think has had a huge impact on the field of cell biology, and it's really helped define modern cell biology in the US and globally.